It's no secret that the world we live in is teeming with innovation in all areas. Each new smartphone, sustainable energy solution, or medical breakthrough seems to push society past where we already are. But a 2019 report from Scientific American tells us that despite what we may think about the amount of progress present in our modern world, the rate of major technological innovations has actually slowed considerably. It could be said, then, that the rate of societal progress in general mirrors the graph of a logarithm, with rapid growth at first, which then tapers off. And this is the story for many fields of research, from aviation to medicine to, as we will discuss today, gene editing technologies. For decades, scientists used various techniques and made several incremental discoveries in their efforts to pioneer an effective gene editing technology. But in 2012, the bombshell breakthrough came after years of stagnated progress, when biologists from the University of California at Berkeley developed CRISPR-Cas9, the most versatile, cost-effective, and reliable gene editing system to date. CRISPR, or clustered, regularly interspaced, short palindromic repeats, was first discovered in nature, where it acts as an immune system for various microbes. But to truly understand how CRISPR works on a molecular level, we must first understand the characteristics of microbial DNA as a whole. In 1987, Japanese researchers observing the E. coli bacteria noticed that a section of its DNA, which holds its genetic information, had two distinct parts. First, they found sections of the DNA that were short and palindromic in nature. These repeats were separated by sections of non-palindromic DNA called spacers. Thus, these palindromic repeats were clustered, short, and regularly interspaced, giving CRISPR its name. As it turns out, though, the spacers are far more interesting than the titular repeats. In fact, the spacers between the repeats were found to encode the genetic information of viruses, which the cell recorded during past infections. And this is how the bacteria protects itself from viruses, by chemically remembering past invaders, just like our immune systems protect us. But how the bacteria recorded this genetic information remained a mystery until 2006, when a protein called CRISPR-associated protein 9, or Cas9, was discovered in bacteria. This protein is extraordinary, since it can cut DNA, hence its nickname molecular scissors. Upon infection, this protein cuts out a section of the virus's DNA and places it in a spacer between two palindromic repeats. Then, if the same pathogen returns to the cell, the match between its genetic code and the code in the spacer generates an immune response which kills the virus and prevents further infection. And although this process occurs naturally in bacterial cells, the team of researchers from Cal Berkeley were the first to harness the power of CRISPR, optimizing it for use in all sorts of scenarios outside of just viral immunity. Most notably, they gave humanity the unprecedented power to edit the genes in nearly every living organism. And for their findings and scientific innovation, the researchers were awarded the Nobel Prize in Chemistry last year, speaking to the significance of their discovery. And now, almost 9 years later, CRISPR is seemingly everywhere. It's now being used to edit the genes in everything from coffee beans to make natural decaf, to wheat to make it organically gluten-free, to even algae to make more clean and sustainable biofuels. But what is undeniably the most important and controversial use of CRISPR is its ability to edit human genes, representing an opportunity to cure previously uncurable genetic diseases. For example, the CDC found that CRISPR could be used to help treat heart disease. Stanford School of Medicine found it aids in treating asthma. The University of Pennsylvania proved its use in treating cancer. And Boise State University discovered its use in even curing Alzheimer's disease. But in today's world, I know we are all wondering, can CRISPR treat COVID-19? And the answer is yes, because CRISPR presents an interesting and brand new alternative to conventional vaccines. It's logical, too, since CRISPR exists for the sole purpose of fighting off viruses in nature. So, to test this theory, researchers from Stanford University used CRISPR in a technique called Pac-Man and they were able to prevent 90% of COVID infections in lab tests. A CRISPR-based vaccine was even considered for mass distribution, but the mRNA alternatives proved timelier to manufacture. But despite the clear power that CRISPR holds, the technology also raises significant ethical questions. And these issues can be broken down into 4 main points. First, a lack of scientific and ethical consensus around CRISPR's use. The National Board of Medical Examiners explains that in regard to CRISPR, the scientists have moved quicker than the ethicists, and as a result, the places in which using CRISPR is acceptable remain largely unknown, because it can be used to prevent fatal diseases, or make food safer, or stop a global pandemic. But it can also be used to edit genes under medically unnecessary conditions, such as altering the genes for eye and hair color, height, or muscle tone. And in the wrong hands, CRISPR could make designer babies and eugenics an alarming reality, leading us to the second ethical concern: thoughtlessness. In late 2018, Chinese biologist He Jiankui proclaimed that his team had created the world's first gene-edited babies, to much fanfare. But it was later revealed that he had done so without receiving proper consent. While Jiankui is just one example, the thoughtless use of CRISPR could present an ethical disaster if left unchecked. And unfortunately, this is happening as a result of the next ethical issue: a lack of regulation for the technology. No single worldwide institution exists for the regulation of gene editing technologies, meaning that the line between acceptable and unethical is currently blurred beyond recognition. And this creates the final ethical dilemma: a cultural unpreparedness. Ethicist Robert Klitzman warned us in 2019 that CRISPR was on its way, but that we were not ready for it. And it's easy to see why he said that. The lack of ethical consensus on the use of CRISPR, coupled with a lack of legal regulation, creates a perfect storm for the technology to be used for detrimental purposes. So, what should we do? Well, CRISPR will keep popping up all over our society, elevating biology, genetics, and science as a whole to a whole new level. But along the way, we need to appreciate but conserve its power. And down the road, with a firmer ethical, cultural, and legal understanding of this technology, we can embrace CRISPR for good.